Good evening, ladies and gentlemen. Just giving, as usual, 1 minute or so. There's still plenty of people joining. Okay, right. I'm going to get started. There are still a few people joining, but I think we can get going. Good evening. Welcome to tonight's CPD presentation from the Center for Human Metabolomics. We have been inviting people external people to join as panelists for the past few weeks, but I didn't invite anyone for tonight. So if you are in the audience and you'd like to join the panel, then please just indicate to me and I will promote you then to panelist, particularly if you can offer some experience with ordering the 6-plex LSD test in both the public and the private sector. Tonight's presentation is sponsored by Pfizer, although they do not influence the content of the presentation in any way, and they also have no influence over the selected topics. There's just one thing that I'd like to mention about the CPD points. So we've had a few hiccups with our service providers, so you probably would notice that not all of your CPD points reflect on the HPCSA portal, but we are in the process of correcting that. They should come in within the next few weeks, and I will inform you when everything should be up to date. So if by then there's still an issue, then you're welcome to contact us, but just keep an eye on that. It will update within the next few weeks or so. Then tonight's presentation is by Dr. Monique Opperman. She earned a PhD in 2023 and is now a postdoctoral fellow at the Center for Human Metabolomics at Northwest University. Her doctoral research offered significant insights into how tuberculosis treatment impacts urinary metabolomes in active TB patients, enhancing the understanding of the disease's effect on metabolic profiles and treatment responses. Currently, Dr. Opperman continues to build on an expertise in TB by employing advanced metabolomics techniques. Her work includes assessing the suitability of the guinea pig as a model for TB metabolomics research. In addition to TB-focused studies, Dr. Opperman is deeply engaged in broader research activities at the Center for Human Metabolomics, contributing to data analysis for various commercial projects. She also collaborates with a multidisciplinary team, enriching her research with diverse perspectives and fostering innovation. Dr. Opperman is expanding her research to encompass inborn errors of metabolism, drawing on insights from collaborative projects. This interdisciplinary approach enables her to integrate various methodologies, furthering the understanding of metabolic disorders and their genetic foundations. Monique, so well done with your studies up to now. We will forgive you for starting in tuberculosis, and we're glad that you made the move to inherited metabolic diseases. Your topic tonight is the diagnosis of lysosomal storage disorders in the laboratory, one of my favorite topics, and also the favorite of, I know, some of the people in the audience. Thank you very much, and good luck.
Yeah. Thank you, everyone. So I hope I do this presentation, this topic justice. So as Prof mentioned, I've came from TB research, so I've done a lot of research on this topic, but please collaborate if you have any insights to add. And yeah, hopefully I can give you a good overview of the most common diagnostic techniques used to diagnose lysosomal storage disorders. Okay, so first off, I'm just going to give an overview on what the lysosome is and what function it has. So the lysosome are responsible for the breakdown and recycling of macromolecules, including carbohydrates, lipids, nucleic acid, and proteins, and it functions as a metabolic hub that controls nutrient sensing, amino acid and ion homeostasis, and calcium signaling. So basically, the lysosome is the digestive or recycling system of the cell. It degrades material taken up from outside or obsolete components within the cell and break it down into smaller components that can be either reused by the cell or excreted. So lysosomes can be compared to a refuge removal system, and lysosomal storage disorders can be compared to the waste management system breaking down. Sorry. So imagine if a garbage truck stops working properly and trash piles up in the streets. Normally, the city's trucks collect these and breaks down the waste to keep things clean. But in LSDs, these trucks are either missing or malfunctioning, meaning the garbage doesn't get picked up. And over time, this waste builds up, causing the city, or in this case, the body, to experience more and more problems. And eventually, parts of the city can no longer function properly. So just like a city needs its garbage removed to stay healthy, our cells need enzymes to break down and recycle the materials. And when that doesn't happen, the accumulation causes serious health issues, as seen in LSDs. So in LSDs, there are various enzymes that need to break down these macromolecules, and if they are not functional, in any case, like enzyme deficiency might occur, then substrates start accumulating. These substrates deposit in various organs, and this causes disrupted cellular function, which causes a lysosomal storage disorder. So for lysosomal storage disorder, there are more than 70 monogenetic inherited metabolic disorders. Individually, they are considered rare, but they have a combined prevalence of about 1 in 5,000 to 1 in 6,000, depending on the country or on the country, basically. So most of these disorders are autosomal recessive, meaning that a child will be effective if both the parents are carriers of this mutated gene, and then some or 3 of these LSDs are however X-linked, like Danon, Fabry, and Hunter syndrome, MPS 2. The following figure briefly illustrates the dynamic state of a lysosome function within the cell. So synthesis of hydrolytic enzymes begins within the nucleus, where genes encode lysosomal hydrolases. This include proteases, lipases, glycosidases, all the enzymes responsible for breaking down these specific types of macromolecules. They are transcribed to mRNA, and these mRNAs are translated into inactive proenzymes by the ribosomes associated with the rough endoplasmic reticulum. In this endoplasmic reticulum, the lysosomal enzymes undergo post-translational modifications such as glycosylation, which is crucial for their recognition and targeting. These enzymes are then transported to the Golgi apparatus, where they are further modified. For instance, these enzymes receive a mannose 6-phosphate tag, which is essential for them to be directed to the lysosome. These lysosomal enzymes, which are tagged with M6p, also bind to M6p receptors in the trans-Golgi network, and then these enzyme receptor complexes are packaged in vesicles that are transported to late endosomes or phagosomes, or the autophagosomes that degrade either the obsolete cellular components and organelles or the bacteria that's engulfed. And these lysosomes bind with these vesicles, these lysosomal hydrolases, the enzymes bind with these endosomes, and then a lysosome is formed, where these enzymes can then start to function to break down these macromolecules into smaller components that can either be excreted or reused within the cell. So the lysosome seems quite not so complex when we look at this figure, but when we take a closer look, we can see that there are very distinct or a lot of stuff going on in a lysosome. So for instance, we have a lot of proteins involved here in the transport, for instance, cholesterol transporters, sugar transporters, diet transporters, amino acid transporters. So there's a lot of transporters, ion channels like calcium channel, ATPase. Then we also have enzyme activators and proactive factors within the lysosome, transport proteins, and acid hydrolases. And this is involved in a lot of functions of the lysosome, including signaling and ion homeostasis, lipid metabolism, the export of products from the lysosomal cargo degradation, gene regulation, motility. So as you can see, the lysosome plays an essential role in normal cellular function. And when it is a defect on any of these proteins essential for the breakdown occurs, it could lead to an accumulation of product, which could then disrupt cellular function. So the following figure illustrates the mechanisms involved in pathophysiology of LSDs. The figure highlights the complexity of LSDs, which disrupt multiple cellular pathways, including transport, calcium balance, and immune responses. The accumulation of undegraded substrate initiates a cascade of events that are increasingly recognized as key contributors to LSD pathogenesis. These include the storage of secondary substrates not directly related to the defective enzyme, impaired autophagy, disrupted calcium homeostasis, mitochondrial dysfunction, and oxidative stress. And many LSDs also involve the dysregulation of signaling pathways and activation of inflammatory responses. In the following figure, it's an indication of the various different LSDs and LSD-associated disorders which are presented. And as seen here, there are a vast majority of different proteins and complexes that can be affected, and this makes the diagnosis or pinpointing the exact defective protein or pathway difficult. To highlight a few of these LSDs in this figure, like mucolipidosis type 2 or I-cell disease, is caused by a deficiency in the enzyme N-acetylglucosamine 1 phosphotransferase. And this enzyme is basically critical for the tagging of the lysosomal enzyme with that mannose-6-phosphate marker, as I've previously mentioned, which directs it to the lysosome. Now, without this marker, the enzymes are excreted outside of the cell instead of going to the lysosome. This leads to severe physical abnormalities, including skeletal deformities, growth delays, and developmental issues. So symptoms usually appear in infancy, and the disease often leads to early death. In Danon disease, as we can see here, there's a deficiency in the LAMP2 gene, and this impaired the normal degradation of cellular waste inside lysosomes, leading to accumulation of cellular debris, and this results in damage to tissues, especially affecting the heart, skeletal muscles, and sometimes the eyes and the brain. Niemann-Pick type disease type C is caused by mutations in MPC1 and MPC2, and this leads to a disruptive cholesterol transport from the lysosomes, causing lipids to accumulate abnormally within the cells, and this leads to progressive neurological and physical deterioration. Continuing on, we also have Pompe disease, known as glycogen storage disease type 2. And this is caused by mutations in the GAA gene. This gene provides instruction for the producing of enzymes called acid alpha glucosidase, which is responsible for breaking down glycogen into glucose. And then I can go on. There are a lot of these defects. For instance, we have cystinosis, which is transport of cysteine that is affected, and mucolipidosis type 4, caused by mutations in the MCOLN1 gene. And so we can continue. But then in the hydrolyses themselves, these hydrolytic enzymes, we can also get mutations, and this, when we zoom in here, we can see it can be characterized as phingolipidosis, glycoproteinosis, mucopolysaccharidosis. So sphingolipidosis or LSD is caused by defects in the metabolism of sphingolipids, which are complex lipids found in cell membranes. These disorders are typically characterized by accumulation of sphingolipids or they're metabolized due to deficiencies in specific enzymes involved in their degradation. Some notable examples are Gaucher disease, Krabbe disease, Tay-Sachs, Niemann-Pick A and B, and Krabbe disease. So glycoproteinosis refers to a group of LSDs characterized by the abnormal metabolism of glycoproteins. And when we look at mucopolysaccharidosis, these are a group of LSDs caused by deficiencies in enzymes responsible for breaking down glucosaminoglycans, GAGs, also known as mucopolysaccharides. So GAGs are long chains of sugar molecules that are part of connective tissue, cartilage, and skin. And when these enzymes are deficient or malfunctioning, GAGs accumulate in various tissues and organs, leading to a range of symptoms and progressive damage. So the intricate nature of these disorders often pose significant diagnostic challenges, requiring a multifaceted approach to accurately identify and manage them. So the diagnostic pathway of LSDs typically begins with the recognition of clinical symptoms that may be subtle or overlap with other conditions. Early suspicion is often guided by a combination of these clinical features, a family history, and potential initial screening tests. Confirmatory diagnosis involves a systematic evaluation, including biochemical assays to measure the enzyme activity levels, as well as molecular genetic testing to identify the specific gene mutations. And this can be accompanied by additional imaging studies to assess organ involvement, etc. So, and then based on all these findings and presentations, you have to confirm your diagnosis. And I will get into that, after which treatment can start. So in this presentation, my main focus will lie on this screening and the enzyme assays, but I'm going to discuss a bit on each of these topics. So for a clinical presentation of LSDs, it is as varied as the disorders themselves, reflecting the complex interplay between enzyme deficiencies, the substrate accumulation, and organ involvement. These disorders often present with a range of symptoms that can affect multiple systems, making early diagnosis challenging. Classic features could include progressive cognitive impairment, developmental delays, neurological symptoms such as ataxia and seizures, coarse features, organomegaly, abnormal eye findings, and skeletal abnormalities. Usually, the substrate accumulation occurs in the organs where they are synthesized, like the liver, spleen, or bone, which partially explains the involvement of all these different organs. And these are just some of the major organs that could be affected with these different types of LSDs, including brain, lungs, kidneys, the liver, the skin, muscles, bone. So it's this clinical presentation does go wide. So when to expect a lysosomal disorder? So a very broad overview is here. These disorders are often not immediately apparent at birth, with most affected individuals appearing normal in the neonatal period or shortly thereafter. The hallmark of these conditions is progressive deterioration, typically without acute exacerbations as the disease progresses. Several key organ systems are commonly affected, and specific clinical findings help point towards an LSD diagnosis. So the central nervous system, patients may experience global developmental delay or regression, often accompanied by epilepsy, movement disorders, or signs of upper motor neuron lesions. In the digestive system, hepatosplenomegaly, enlarged liver and spleen, is a common feature which may not or may be associated with cytopenia, a low blood cell count. Musculoskeletal system, facial dysmorphology, distinctive facial features, and this dysostosis multiplex skeletal abnormalities are key indicators, and ophthalmological findings are related to issues such as clouding of the corneas or other specific findings are frequently observed. While classic presentations are well known, LSDs can also present in attenuated forms or as congenital disorders where symptoms manifest early and severely. Certain features such as progressive organ involvement help identify specific orders. So LSD present with a continuum of disease severity that loosely correlates with the type of mutation and residual activity of the mutant protein, but they are generally classified on the basis of the type of disorder, which are determined by the type of enzyme that is deficient, the accumulated substrate, clinical presentations, and the mode of inheritance. The age of onset of the clinical signs is also used for the classification as congenital of infantile, which are most often the more severe type, then late infantile, juvenile, and adult types. So in this presentation, it is important to acknowledge that there are numerous screening and diagnostic assays available for LSDs, and I won't be covering all of them since various countries and laboratories it differs and varies between them, but I'm just going to focus on those mostly done in South Africa and going to go into this. So when looking at preliminary screening tests, there are several effective screening strategies available for different classes of LSDs. So for MPS disorders, for instance, a urine glycosaminoglycan, a urinary gag test is widely used. And in the case of urinary oligosaccharide, in the case of oligosaccharidosis, urinary oligosaccharides are usually screened for, and I'm just going to go into that a bit more now. So first, looking at the gag test. So the basic principle of these urinary gag tests is to look for the presence of different glucosaminoglycans in the urine. GAGs are negatively charged, long, unbranched polysaccharides consisting of repeating disaccharide units with variable sulfation levels. So here is just a group of the 5 major groups of the GAGs and their repeating disaccharide units. So in MPS disorders, there is a defect in the enzyme responsible for the degradation of these GAGs, leading to their accumulation in tissues and increased excretion in urine. The analysis aims to detect these abnormal levels or patterns of GAGs in urine. So the detection for abnormal excretion patterns of GAGs can be done on multiple ways, and here I'm first going to discuss the electrophoresis of urinary GAG screening method. So first you have your urine sample. You will prepare your buffer and gel, your gel or membrane preparation, prepare your urine sample and your GAG standards, load the samples and standards into the well of the gels, and then run the electrophoresis, stain the gel or the membrane, destain this, and just important here to note, so although you destained the gags that you, the gags will retain the staining, and then you will be able to visualize and interpret the patterns of the gags using a UV transilluminator. So for this, you will compare the migration patterns of the patient samples with those of the standard gags. With this, you will identify abnormal gag patterns based on the differences in the band positions and their intensities, which can indicate the accumulation of specific gags associated with different MPS disorder. So for the interpretation of results, so normal samples would typically show little to no visible staining, as normal individuals excrete very low levels of gags. Abnormal samples will show distinct stain bands that correspond to various gags. And for instance, these patients with dermatan sulfate gags detected could be suggestive of an MPS 6, but confirmation with enzyme assay is needed. Then patients with dermatan sulfate and heparin sulfate will be indicative of MPS type 1, 2, and 7. And then for keratin sulfate, we will look at these MPS types. So as I've mentioned, there are a lot of ways, and that is just the electrophoresis type, but these urinary GAG analysis can also be done on an ALSI method, and this is also what is used at CHM for the urinary GAG analysis. This is a more quantitative and a bit more reliable than just to compare this visually. You detect it, and then these GAG detection or the presence of the GAGs can be compared to other profiles to determine if it is irregular. Next up is the oligosaccharidosis screening, where you will look for an excess of oligosaccharides in the urine. So the deficient enzyme inhibits the catabolic pathway and results in accumulation of non-degraded oligosaccharides in the lysosomes and body fluids. The composition of these non-degraded chains depend on the specific enzyme deficiency. Therefore, these chains are good biomarkers for the rapid identification of a candidate gene that causes the disease. So figure 1 shows the mechanism by which the lysosomal enzymes degrade N-linked glycan. So here you can see each enzyme is responsible for the degradation of the shortening of this oligosaccharide with a certain protein, and this causes changes in the size of the oligosaccharides that's detected. And when we look at the method, typically thin layer chromatography is still used here. So you will have your urine sample, you will prepare your TLC plate with your control samples and space for your patient samples. Spotting of the sample and the standard occurs, and then you prepare your solvent and also develop your TLC plate in the solvent, that with capillary action, the solvent will move up, and then you will stain and heat the TLC plate to get the color changes or just to develop the colors of the migrated oligosaccharides and interpret the results. So here you can also use the retention factor to compare the movement of the detected oligosaccharide with your standards. So for the urinary oligosaccharides, typically here are a few disorders, the deficient enzyme that will be investigated, and the accumulated substrate that will most probably be what you're looking for in your TLC plate. And again, this will be confirmed with, these patterns will be confirmed with other standards to determine what abnormal pattern was detected here. Then, although there are reliable screening tests available for the different LSDs, essential, sorry, let me just laser out, just want to remove that. Okay, so although there are reliable screening tests available for different LSDs, confirmatory tests are essential for accurate diagnosis and management of the condition. These include enzyme assays that could be done using fluorimetric assays or tandem mass spectrometry. Enzyme assays for LSDs can use a variety of sample matrices, including blood, plasma, urine, and fibroblasts. Each matrix offers unique advantages depending on the enzyme being tested. For instance, plasma and urine can be useful for detecting enzyme activity relating to certain storage products, while cultured fibroblasts may be used when more specialized or confirmatory testing is required. However, dried blood spots are one of the most common sample types used for initial LSD screening and enzyme assays due to their convenience and reliability. So dried blood spots are collected by pricking a finger or heel to draw a small amount of blood, which is then blotted onto the filtered paper. These spots are easy to collect and to handle, making them widely accessible method for testing. There are various sources that can, like also mentioned in a previous presentation, you can just Google or YouTube a method to see how dried blood spots can be collected easily. But also when you request a test, there are also collection kits that are available from different laboratories, such as from CHM, which gives you clear instructions on all the necessary materials, the proper way for sample collection and transport, the drying process, storage conditions, and also any handling requirements to ensure high quality sample for testing. So this simplifies the process for both clinicians and patients, ensuring reliable results across different testing needs. So for enzyme analysis, we will actually need a 3.2 millimeter punch from these dry blood spots, which will be used to extract enzymes and then to test their activities. So considering that we need a punch, basically a whole punch from these dry blood spots, high quality samples should be ensured. And this is why there's also instructions with it, because let me just show in a few examples where collection is not ideal. So when the cords are not fully saturated in the filter paper, it means that there is not enough blood, or so when you punch out a dry blood spot and there's not, the whole punch is not filled with blood, it can influence your readings and maybe lead seem like deficient enzyme, but actually there was just less blood analyzed in this moment. So that's why you should ensure that the whole circle is filled. And then specimens should not be scratched or abraded. They should be fully dry before mailing. Any specimen that appears super saturated, diluted, discolored, or contaminated, exhibits serum rings, or appears clotted or layered may influence the analysis. Okay, so for plurimetric assays, let's start with that. With these assays, you want to detect the enzyme activity. Now, your enzyme of interest will have a specific substrate that links to it. And then for these fluorometric assays, we take this substrate that would link to an enzyme, and this is linked to a fluorescent product or substrate. So 4-MU is in this case. And the principle here is that when this linked fluorescent product with your substrate binds to the enzyme, this fluorescent product will be released. And this reaction is only going to occur when the enzyme activity is actually is functioning properly, or if there is enough enzymes for this. So this fluorescent product is directly correlated to your enzyme activity in this case. So the higher the enzyme activity, the higher the fluorescence emitted. So when there is only little of this enzyme in your patient sample, or if this enzyme is deficient in some way, this reaction won't be able to take place, and this will result in low enzyme activity, correlates to low fluorescence. So that's just the principle of this fluorometric assay. And here you will use your dried blood spot, which will be punched in a 96 well plate, which will also contain your QC samples, your calibrators, blank samples, the whole run. You will add your specific reagent and substrate, incubate the reaction, terminate the reaction with a glycine carbonated buffer, and then you will read the fluorescent activity at different wavelengths and compare these results with that determine the enzyme activity with that of reference ranges. Here I just made a list of some of the 4-MU substrates used. So again, it's the LST disorder, the deficient enzymes, the products or substrates quantified, and the 4-MU substrate used. Okay, so tandem mass spectrometry or MS-MS analysis and analyze the enzyme substrates and products by measuring the mass to charge ratio. In LSD, the MSMS can detect the accumulation of specific biomarkers or unmetabolized substrates that result from enzyme deficiencies. This technique involves multiple stages of mass analysis to precisely identify and quantify these biomarkers. So here I just use the LSD 6-plex as an example to illustrate the method. So again, you have your dried blood spot card, you punch your samples and your QCs and calibrators, and then you add your cocktail mix to extract the enzyme. It's incubated, the reaction is quenched, and then transferred. Your flow solvent for the LCMS run is added, and then you do your LCMS analysis and get your mass spectra and chromatographic results. So for this specific LSD 6-plex test, there is the inclusion of 6 different types of LSD that are tested for simultaneously. So this is an advantage of the tandem mass spectrometry. You can detect multiple or can test for multiple enzyme deficiencies at once. So this is the different tests and the deficient enzymes that are evaluated here. So this is just some of the results. So this is a chromatogram that will result from that LCMS analysis, and the enzyme activities are calculated by quantitatively analyzing a specific product generated when an enzyme reacts with a synthetic substrate. So the enzyme activity in this case will be the area of the product divided by the area of the internal standard multiplied by the volume of the internal standard used, and this whole divided by the dried blood spot volume and the incubation time. So this method, okay, so then you get your enzyme activity, and this this activity value will be compared to reference ranges to determine if this enzyme activity is normal or maybe deficient. So this is a cost-effective approach, and with an improved turnaround time. It allows for multiple enzyme testing instead of only a single enzyme. And another benefit is if there is low enzyme activity or 2 or more enzymes, it may indicate a compromised sample and an immediate repeat can be requested. So a molecular study is then also required for confirming the diagnosis of an LSD that can help in identifying the carrier status in other unaffected members in the family. So Sanger sequencing and next generation sequencing are the 2 I'm going to go in here. So Sanger sequencing, also known as chain termination sequencing, reads the DNA sequence of a specific gene. It involves incorporating labeled nucleotides that terminate DNA synthesis at specific points, allowing the sequence to be determined by electrophoresis. So yeah, the process, you can use dried blood spots in this case. I just indicated the blood sample. You do your DNA extraction. You mix the DNA and reagents, do a PCR amplification, capillary gel electrophoresis and fluorescence detection, and sequence analysis and reconstruction. For next generation sequencing or high throughput sequencing, it analyzes the entire genome or specific regions of interest simultaneously, and it uses parallel sequencing technologies to provide a comprehensive and high resolution genetic information. So again, you have your DNA extraction, your library preparation for the multiple DNA regions you might want to investigate. You do your sequencing, and then you get your result, which then go through data analysis, bioinformatics to try and identify the variants that might be present and causing this lysosomal disorders phenotype that you are seeing. Here is just an example of the disease-causing mutations in MPS disorders. So you have your different disorders, the gene that is affected, where this gene is located on the chromosome, and then pathogenic variants reported. So as you can see here, there are hundreds of pathogenic variants for several of these MPS disorders, and these mutation types can vary from small deletions, insertions, indels, splicing, mutations, gross insertions, gross indels. So there's a lot of different variants that can occur. And just another example of this is in a paper from the Lancet Regional Health, they illustrated in an Indian population just the distribution of the different variants for patients diagnosed with Gaucher disease, and then also in Tay-Sachs disease. So after screening, enzyme assays, and genetic testing, the best approach to confirm a diagnosis, particularly for lysosomal storage disorders or genetic conditions, involves a combination of the clinical evaluation, the test, and the sometimes additional genetic analysis is also required. But considering you can identify a variant of unknown significance, pathological significance, it is important. Well, it can make the whole process difficult. So a few things worth considering in your approach includes to ensure that a patient's clinical presentation aligns with the suspected disorder. You assess the family history for patterns of inheritance that match the suspected disorder. You could repeat enzyme assays and confirm the results by repeating a test or measure specific biomarkers or substrates that accumulate in the suspected disease. You could do confirmatory genetic testing, confirm initial detected pathogenic variants by sequencing an independent sample. Segregation analysis could help confirm the diagnosis, especially for recessive conditions where both parents should be carriers. mRNA functional studies can be done in vitro. Functional studies could be considered. Imaging studies, if that was not already done, like mRNA of the brain, liver, spleen, can reveal characteristic findings such as organomegaly, bone abnormalities, or brain changes that are consistent with the suspected disease. Electrophysiological tests for disorders that affect the nervous system, such as Gaucher or Fabry. Tests like nerve conductive studies or EEG may provide additional evidence. But most importantly, is to consult a multidisciplinary team. So engage a multidisciplinary team, including geneticists, metabolic specialists, and other relevant healthcare providers to reveal all findings and confirm the diagnosis. So when looking at the diagnostic approach in South Africa specifically, we will have a phenotype description, which can either be where an LSD is suspected or a no LSD is suspected or non-specific clinical information is provided. So by first looking at where there is not a specific clinical information, you will do a routine metabolic screen, and this will include your urinary gag analysis, oligosaccharides, and other tests like organic acid, amino acid, and acylcarnitine analysis. These urinary gags and oligosaccharides might indicate that some LSD could be suspected. And then if this is the case, then it is redirected to this route where an LSD is suspected. Now, first off, if a specific LSD is suspected, you will be directly referred for enzyme assays, which could include the LSD 6-plex, MPS2 for males, or secondary biomarker monitoring like lyso-GL1 or GL3 maybe. This will only to be confirmed with genetic tests. And again, if a non-specific LSD is suspected, you will do routine blood work, including your liver function test, creatine kinase, complete blood count, renal functions, urinary gags, and urinary oligosaccharides, which again might be indicative of a specific LSD for which a recommended enzyme test based on the findings can be recommended, and then followed by a genetic confirmation of the test. When specifically looking at the 6-plex LSA approach that is followed, when this test indicates any positive results, genetic analysis will be done to confirm this diagnosis. And also for Fabry, a GL3 test will be requested, and Gaucher GL1. And for MPS, additional urinary GAG analysis will be done. When this 6-plex LSD might be negative, but there is a suspicion of Fabry in females, GL3 will be done. Will be analyzed for MPS. An expanded MPS panel will be done, and also urinary GAGs and ligas will be requested. And for Gaucher GL1, if any of these results are positive, they will also be confirmed with genetic analysis. So as seen here, the 6-plex LSD as well as the urinary GAGs and oligos are currently performed at CHM, where the rest of these tests are performed at the Archimed Life. Just a quick case study. I'm just basically to show what type of results. So clinical information for this case study is a 1-year-old, hypertonia, hepatosplenomegaly, and anemia was presented. The LSD 6-plex test was done, and deficient enzyme activity indicated Gaucher disease, which were then confirmed with a lyso-GL1 test, and then genetic sequencing also indicated abnormal multiple mutations. And here are some of the mutations that were identified, and there were some novel mutations, but also common mutations. So just some statistics. This is not the new statistics, unfortunately, that I have, but just to give an example of what this approach had found already. And from 2019 to 2022, you can see that 84 patients was diagnosed using this approach, and which of the most prevalent was of this group was MPS II and Gaucher disease. Okay, so once a diagnosis of lysosomal storage disorder is confirmed, treatment initiation could commence to try and alleviate some of the symptoms and help these patients deal with the disease. So considering the clinical and genetic heterogeneity seen in LSDs, there have been great challenges in establishing therapeutic approaches for LSDs. Nevertheless, with continuous efforts and advancements in this area, a number of strategies have been developed. These could include enzyme replacement therapies, substrate reduction therapy, enzyme enhancement therapies, and gene therapies. Now, the most successful approach to date is enzyme replacement therapy, which have been applied to Gaucher disease, Fabry, Pompe, some MPS types, and then also Niemann-Pick A and B. In general, the clinical outcome is optimized when treatment is initiated as early as possible and ideally before the development of some of the symptoms. So the key message here is that early and accurate diagnosis of lysosomal storage disorders is critical for effective treatment and improved patient outcomes. Combining the advanced techniques like enzyme assays, mass spectrometry, and genetic testing allows for the precise, timely identification of LSDs. Implementing a multidisciplinary diagnostic approach can drastically reduce the delays and misdiagnosis, helping guide appropriate therapies and improving quality of life for these patients. Here I only added a screenshot of the National Metabolomics platform website of the diagnostic page, where you can get more information regarding these diagnostic services that are available, including the PLSDA, the GAGs, and the oligosaccharides. And then they also have a contact form if you have any alternative requests or want to get more information on some of the processes. Okay, and this is just some of the references that I've used. And with this, I thank everybody for your time, and I hope I could give you some information regarding the lab approach to diagnosing LSDs.
Thank you, Monique. Wonderful presentation. Nice blend
Thank you.
of the analytical, biochemical, and clinical aspect. I'm
hope
pretty
so.
sure it satisfied everybody's
Oh, I
taste
hope
in
so.
the audience. Wonderful presentation. Well done. Okay, I just quickly want to repeat a few things that I said in the beginning because more people have joined us. Please note if you find that some of your CPD points are missing. We've had a hiccup with our service provider. We are in the process of sorting it out, and your CPD points should reflect within the next few weeks. I will inform you when the inquiry is done, and everything is not. If there's something missing, then you're welcome to contact us. Just also, this presentation is sponsored by Pfizer, but they do not have any influence over the content. They simply wish to promote education in rare diseases in South Africa. You are very welcome to post questions in the Q&A session. And also, if you'd like to be promoted to a panelist, then I'd actually appreciate that. You can join the discussion. So while we wait for the questions to come in, Monique,
Yes.
as you know from your reading probably is that, there's been a lot of development in testing for lysosomal storage disorders. I think we've made a lot of progress, but we certainly are not where we can be. So I think we've got a couple of options here. One is to improve our metabolite test. So the glycosaminoglycan test that we use is pretty advanced, but there are better options now. The oligosaccharides, there are certainly more sophisticated options, and there's also the field of glycomics. So that's an option. The other option is to expand the number of enzyme tests that we offer, and then the other option is just to go the genetics route as a primary diagnostic route. So what do you think? How should we approach this?
So I think it is. So specifically when I read up about it, I did wonder about, for instance, oligosaccharides. That is the thin layer chromatography pathway. And I have seen that there are a lot of processes that actually also use an LC-MS approach rather for this. And I think it is dependent on the resources available, of course, but it will be advantageous to actually get some of these tests that can be more high throughput and where it does not necessarily take a few days or hours to develop and all that. But it is complex because there is a lot of LSDs, as we've seen here, and I think it will just be good to actually maybe, and I'm not sure how to get an indication of the prevalence of what LSDs could be beneficial for instance, the African continent, and develop some of those tests. But yeah, I'm not sure. There's a lot of avenues to explore, but it's dependent on the resources that we have available.
Okay,
and time to develop these methods.
Yeah, it takes time.
Mm.
Okay, we've got a couple of questions. Kagiso is saying, great presentation. Thank you, Monique. Mandisa, hi everyone. Nice presentation, Dr. Opperman. Ilse de Priya, how much does the LSD test cost?
Okay, so currently with CHM, the LSD 6-plex test is a free service. So if an LSD is suspected, this test can be requested, and it is currently free, and it's for state and private patients, as far as I understand.
Yes, that's correct. So private patients can just go to any one of the path labs and request the 6-plex LSD. The state patients, a little bit more tricky. It's best to get in contact with us or Ansi Mini actually directly, who will organize the transport of the kit to the patient, and then also collection and transport to our labs. Hopefully in future we can also set it up with the NHLS so that it can actually just be requested through the NHLS. So I'm hopeful that we can do that. But any patient in the country can receive a 6-plex LSD test at no cost. Of course, we have to do this in a responsible way. I'm always for responsible testing. But certainly if you do have a suspicion, then if you're unsure anyway, contact us and let's make sure that we can get the patient tested. Then Eliska Jacobs is saying, I attend for the free CPD points, but have also been involved in growing fibroblasts for your testing at some point in my career. It was lovely to learn about LSDs today. It was comfortable to listen. I couldn't agree with her more. Nice presentation, Monique. I don't see any other questions, strangely enough. There was another question that I was thinking that I should ask you, but I can't remember now what it was. What I wanted, it was perhaps more of a comment. So that case study that you showed, maybe you can go back to that slide that shows the dried blood spots.
I think definitely Prof can elaborate on that case study. Let me just get the right thing to share. Just gonna go back. This one.
Yeah, that's it. Yeah. So you know, if I think about that slide that you showed earlier on about the criteria for the sample collection, then it doesn't seem like these spots meet the criteria. In fact, they look pretty terrible, you know. So should we not have rejected the sample?
I'm not sure. So I think yes, I don't know. Prof can elaborate on why you did not reject the sample, maybe.
Well,
or
the
was
truth
this
is
one
I
of the?
I did reject the sample.
Oh, okay. So
And
it
then
was
the
a
clinician
new
contacted me and said that you know, there's a very high suspicion of Gaucher disease, and can we please run the sample? And then I said, yes, sure. And in fact, what you're seeing there is severe anemia. And that's why those spots are looking pinkish. I thought, you know, the sample was taken from a drip arm or something along that line. You know, so one must always be careful with sample evaluation. And in this case, I actually made an error. Fortunately, the clinician was sharp and informed me that, you know, yeah, that you know, the sample was corrected. So I'm just, you know, always, you know, think about lots of things to think about the lab when evaluating samples and everything that goes with that. So there's a little bit of more comments here. James Sibia saying wonderful information. Thank you, Monique. I must perhaps say that with Monique's permission, we'll make this talk available on our YouTube channel. And yeah, I think it's a great reference for people who want to start with lysosomal diseases, whether you want to look at the clinical, biochemical, and or analytical aspects. And I think regardless of where your focus are, you need to know a little bit about the others as well. That really helps. Ilse Prius asking, what is the different info you will get from GAGs or urine versus DBS? Yeah, that's a nice one.
Different info you will get from GAGs on urine versus dry blood spots. Yes, sorry, I am not sure about that, but let me refer to my own slides quickly. Okay, so I know we do the urine test. So GAGs.
So I like just to think about these things, starting with the mucopolysaccharidosis, because they're very well. They tend to be clinically, although they're also attenuated forms. Usually, the clinical suspicion of a mucopolysaccharidosis is usually very high, with their very distinct facial features and so on. So, you know, the glycosaminoglycans is the test that almost exclusively tests for the mucopolysaccharidosis, and then the enzyme testing is, of course, dependent on which enzyme it's set up for. So in our case, we can do enzyme analysis for MPS 1, 2, 3B, 4A, 6, and 7. You know, but yeah, it's a specific per enzyme. You know, if you have a patient then where another lysosomal storage disease is suspected, then usually they either have a neurological presentation or one of the more specific presentations, like in the case of Pompe or Fabry disease. If it's neurological, you're probably going to start with the 6-plex LSD, but you would also request the oligosaccharide analysis, which can detect some of these disorders, but not all of them. So recently we had a case where there was a high suspicion of a mucopolysaccharidosis. It wasn't the case. We tested the oligosaccharidosis, and the patient had alpha monoacidosis. But if you request the GAGs, the oligos, and the enzyme assays, then you don't exclude all of the lysosomal storage diseases. So if the clinical suspicion remains high, then it's best to contact us for discussion to see what else we can offer to make the diagnosis. Okay, right. I think we have exhausted the questions. So Monique, thanks again. Great presentation. Thank you to everyone in the audience for attending. And we certainly look forward to seeing you next time, and good night.
Good night, everyone.